There is an enemy in nature that is difficult to cure, the bacterium. In nature, however, there are also so-called natural predators of bacteria, that are bacteriophages. But who are these creatures and why are they so important today? Let's find out together in today's video. Hello and welcome back to Med4Care. Today's topic is bacteriophages. What are bacteriophages? Do you know viruses? They are tiny biological machines programmed to infect and exploit the cell's genetic duplication apparatus to replicate. Without cells, viruses could not survive. We are well aware of viruses that are specialized in infecting cells in the human body, such as the flu or HIV virus, or coronaviruses. Well, in addition to them there are also viruses specialized in infecting bacteria. Their name is bacteriophages, literally translated to bacteria eaters. The presence of bacteriophages in nature is impressive. They are ubiquitous in terrestrial ecosystems, especially in seawater surface microbial mats, forests, and agricultural land. Furthermore, bacteriophages establish a record also in numerical terms. In fact, it is estimated that they are the most abundant type of microorganism in the biosphere, with an estimated number of 1,031 types of bacteriophages. There are billions and billions of bacteriophages in every terrestrial ecosystem, so their classification can be complex. Simplifying, bacteriophages can be classified mainly on the basis of two characteristics: their structure and the type of nucleic acids they contain. Structurally, most bacteriophages resemble the shape of small spaceships designed to land on the surface of bacteria to infect them. They are formed by a protein head that stores and protects genetic material, a tail of variable length, consisting of a hollow tube in which the genetic material passes, terminal fibers associated with the tail, which resemble legs and which are used to recognize the bacterial surface and anchor to it. The tail, together with the fibers, forms the so-called injectosome, for example the apparatus which first drills the bacterial surface and then injects the genetic material inside the bacterium like a syringe. More than 95% of known bacteriophages have this tail. For this reason their order in nature has been called Cordovirales, and contain double-stranded DNA as nucleic acid. Depending on the type of nucleic acid, bacteriophages can contain both single and double-stranded DNA or RNA. Their size can vary widely, ranging from the smallest 20 nanometers to the largest such as T4, which measures 200 nanometers in length. Now let's see how bacteriophages replicate in bacteria. Each type of bacteriophage specializes in infecting only one type or family of bacteria. This is because the bacteriophage only recognizes the unique structures of its bacterial target by the tip of the tail. When this interaction is productive and correct, a series of events are triggered which culminate in the injection of the phage genome into the predated bacterium. After the insertion of the genome, and sometimes of accessory proteins, the bacterium can meet two fates. In the first case, the virus takes control of the bacterial genome's reproduction mechanisms and transforms it into a bacteriophage factory. This type of infection usually ends with the assembly of many new bacteriophages within the bacterium and ends with the lysis of the same so that they are released into the environment and infect other bacteria. This type of life cycle is called the lytic cycle. Wait a minute, do we all know what lysis is? It is the dissolution of a cell by various agents. Alternatively, some bacteriophages do not immediately kill the bacterium they infect but insert the genome inside, so that it replicates with it. The virus then spreads covertly in the genome of the infected bacterium's progeny until a triggering event stimulates bacteriophage production and eventual lysis of the host. These bacteriophages are defined as temperate, and the cycle is called lysogenic. Now that we know better the bacteriophages, let's see what are the applications of bacteriophages. The World Health Organization has dubbed this historic moment the post-antibiotic era. In this scenario, bacteriophages may constitute a promising therapeutic alternative to ineffective antibiotics. However, their use does not end here. there are applications in the rapid identification of bacteria, pollution, food preservation. In fact, they can act as biosensors of bacteria in foods, biological control agents in agriculture and aquaculture. Given the inexorable spread of antibiotic-resistant bacteria, bacteriophages are reawakening interest as natural antibacterial agents that could be used as an alternative to or in combination with antibiotics. Bacteriophages prove to be very useful for detecting bacteria and as a method of controlling bacterial growth in food. To make the presence of toxic bacteria visible and measurable through bacteriophages, so-called biosensors have been constructed. Not only in food control, but bacteriophages are also useful in the previous steps of the production chain, agriculture and aquaculture. The use of bacteriophages can reduce the use of antibiotics in agriculture, increasing the yield of the fields, reducing the environmental impact and favoring antibiotic-free products. Furthermore, specific bacteriophages can be used to treat some bacterial diseases affecting farmed fish for food use such as sea bass, trout, sea bream and salmon. At this point, I'd like to know how bacteriophages come to be discovered. In 1917, Felix de Hrull noted that dysentery bacteria were lysed by a cultured agent forming plaques in his plates, and that this agent continued to spread even when diluted. Then Felix understood that there was an invisible microbe capable of parasitizing and killing the bacterium. After excluding that the bacteria were eliminated by other agents, Felix de Hrull was able to give the name of bacteriophages to these microbes. To get the right confirmation of his theory, he even consulted an absolute authority like Albert Einstein. In 1935, Wendell Stanley in New York gives a change to the research on bacteriophages, completely excluding that they are inanimate chemical agents. He succeeds in crystallizing the tobacco mosaic virus and finally proves that viruses are composed of biological macromolecules, such as proteins and RNA. For these experiments, Stanley was awarded the Nobel Prize in Chemistry in 1946. Now it's time for the big dilemma. One of the questions that most intrigued scientists in the years of the discovery of bacteriophages has an almost philosophical undertone. Are bacteriophages living beings? Some scientists define life by the ability to replicate itself and by the ability to be born and die. Viruses are parasites wholly dependent on the cell they infect, so the general idea is that viruses are non-living parasites of living metabolic systems. But the debate is still open. To conclude, bacteriophages are ancient biological tools, widespread in every terrestrial ecosystem, which allow the containment of the various bacterial species. Their study offers us interesting ideas to understand and continue the fight against bacteria. To learn more about the topic in our portal, click on the link in the description. If you found it useful, support us with a nice like, subscribe to the channel and click on the bell to be notified every time our new video comes out. See you soon on Med for Care.